Pohihiri po rārama po te whakaaro po te tangata po te aroha te pauahere nei tātou mai riora kia tātou haumia hui e tāe ki e. Tēnei no te mihi a koutou I tai mai tēnei ahi no reira nau mai haramai ki tēnei o ngā hui o te Whāki. Just saying welcome introduction to everyone that's come along this afternoon to listen to this Whāki webinar. Just a couple of little, I guess a couple of things. So the focus of this is around digital sequence information and benefit sharing. I'll give a little bit of a background to how it came about. All of the presenters here today, Katie Riddle, Tim Coltman, myself and Jacob Golan are a part of our research project called Tikanga and Technology. Has a focus on Indigenous, or at least our component of the project has a focus on Indigenous data and systems, and thinking about how Indigenous data could be recognised, but also how it can be valued. And that's where these different, the context for this discussion has emerged. If people would be good enough to add their do the introductions in the chat, just name and where you're from, that'd be great. We can stay in touch with you if you leave an email there as well. And look forward to spending 45 minutes or so with these 4 presentations and then doing some Q&A at the end. So with that, I'll pass it over to Kaylee. Kia ora koutou.
Oh, kia ora. Let's share screen. Right. So kia ora tātou, ko Katie Lee toka ingoa, ko rongo whakata toka iwi. I work here at Te Kotahi Research Institute, specialising in multi-data sovereignty and intellectual property. And through that, I've been doing a bit of mahi within DSI. So I wanted to talk a bit about IPLC, or Indigenous peoples and local community rights within DSI, and where that space is currently developing. So first of all, just to recap what DSI is, for those who don't know, it's the dematerialized and digitally stored versions of data based off of DNA and RNA. And this can be done from multiple different sources, multiple different samples, or single source origin. And it has a huge amount of value within research, so especially in public health data, medicine, biodiversity, plant animal breeding, and evolution research. And it's been helping us make huge technological advances worldwide. So it has a huge amount of value, and just to see a bit of its pathway from its use. So you'll see here on the left, we have a genetic resource, the source origin, and from their researchers done upon it. So they unwind the strand, they sort it into base pairs, so A, T, C, and G. And you can also do it with protein, and that gets unraveled into a code and put into a data bank that can be open or closed source. And from their research is done on it, and you can use it to generate commercial values, products. And it has so many different kind of uses that it goes into, be it just research, product, commercialization. But the part that I am particularly interested in is benefit sharing and the prior informed consent, mutually agreed terms, and how communities and researchers and scientists, etc., start to have these conversations around benefit generation before we get to the research or the commercialization. So currently within international law in access and benefit sharing currently only relates to physical genetic resources, so not including human resources. So that means that because it's not a physical genetic resource, DSI currently isn't covered within the ABS mechanisms currently available. And so when this DSI gets put into databases and data banks, these are often open source or open access. So they are readily accessible by scientists and researchers, or you might need a membership to access them. And this, as a result, can leave a lot of communities out because they're not having these conversations with researchers and scientists saying, okay, we're comfortable having our data used for this, or the samples generated from our whenua, we don't want it to be used this way. So having those conversations being able to be fostered is really important. And so this kind of leans to the IPLC mindset of as open as possible and as closed as necessary when it comes to open access in databases. because we don't want our traditional knowledge and our taonga species to be taken advantage of or to be used without having a conversation with us as community first. So also as part of that, DSI, as it sits in the database, there is associated metadata. So metadata is data that is about data. So here's the sample, here is the DNA sequence or the DSI. Here's where it's collected, when, where it's from, who's associated with it. So that metadata for us is really important because there you can have your provenance information. This is where it's collected. This is the community associated with that sample or that DSI. And so in the future, when it goes onwards for more usage, you can look in that information and say, this is where it's from. This is who I need to talk to. So having provenance information for IPLC and for benefit sharing in the future is quite key for us. So at the moment, that's currently not often the case. And if so, I think it's about 6% of DSI has associated provenance metadata attached to it, so not a lot. And often that might not be at all associated with Indigenous lands. It's just where in the world it was taken from, or the source country, which is hugely broad. And for us in New Zealand, that doesn't take into account which iwi it belongs to. So it's just this sample is from New Zealand. And that for us isn't enough. So for us, where does our traditional knowledge fit? Where does our mātauranga fit? So the Nagoya Protocol, which is currently in place, doesn't cover DSI, but it does, however, look into the Indigenous relationships with genetic resources. And this is recognized through associated traditional knowledge. So from communities, Indigenous relationships to genetic resources are maintained both by their relationship with the whenua and the land, and also through their mātauranga or their traditional knowledge. So these two aspects are showing the Indigenous connection to the samples that are taken. And so when samples are taken, genetic resources are made, and DSI is generated from that, you can see that there's a link there. So there's the community, there's their whenua, there's the sample, there's the generated DSI. And that to us shows a whakapapa link. And so that relationship there shows that it's important to recognize our relationship with the DSI in that sample, and also with our traditional knowledge related to that particular plant or species, or whether it be a taonga species or something generated from our land. So for us, recently, COP15 was looking at biodiversity, and that was held in Montreal in Canada. And at that conference, they were talking about DSI and its place in international law and what it should be covered by and how that should be governed. And then also, as Tim will also be discussing, we did a valuation of Māori data and DSI within that. So we've got a couple of different things rolling around at the moment. I'm going to talk about COP15. I was there, and a lot of our attendees to the Whāki webinar will have also been there. So if this is already relevant to you, well, so here is the outcome document that came out of that. There's a link for that available there. I just wanted to show you what the cover page looks like so you're familiar with it. And it's still a draft decision. It's not officially in place at the moment. So it's important to remember that this is still a working document, and these are still provisions around DSI that are still being discussed and still being evaluated. So we don't have any concrete international law about DSI yet, but this is how it's starting to shape up. So for us as Indigenous peoples and IPLC, there is a couple of really key principles there for us. So first of all, having the fair and care principles in there, we've discussed those in previous webinars. Those are about having fair and proper adequate provisions in place when it comes to research. And so for us, the F in that is findability. And that speaks to the provenance metadata. In my opinion, it's a way of acknowledging the F in that is to have adequate metadata associated within these samples within the databases. However, you'll see that, where is it here? Point number 4, they're talking about encouraging provenance metadata to be within the public databases. So that affirms that F within the fair and care principles. However, you'll see in point 5, it's juxtaposed by saying that tracking and tracing of digital sequence information isn't practical. Now, while these aren't tracking and tracing and provenance metadata aren't the same thing, they have a very similar link. So there's a bit of confusion at the moment about where does the line end with tracking and tracing, and how much information do we need to have on these databases. So that's currently one of the soft points in the developing framework at the moment that we'll see being discussed over the next couple of years. Also, there was an agreement that we need to have an adequate access and benefit sharing mechanism in place for making sure that communities are getting fair recognition and fair benefit back from the utilisation of their resources. So if you see in point 9I, it very specifically says that there needs to be provision for Indigenous peoples and local communities, their traditional knowledge or amatōranga, and with the genetic resources they hold. So with our taonga species and our amatōranga and the specific cultural rights and interests that we have in our whenua. So from there, there's also going to be, or you'll see in points 10 and 11, this is also enforced as well. And it needs to not affect the Nagoya Protocol, and it needs to be working with what's already in place. So considering the rights that we already have under the Nagoya Protocol, we're hoping that these are going to align really seamlessly. So a couple of key points within that, like I said, there's currently a bit of confusion around provenance versus tracking and tracing and where our obligations lie. And we would argue that it's really important to have proper, adequate provenance metadata, which will be discussed and seen in the Manaaki Whenua example. So you can see that in action and how that works. And then also there's discussion of what does the benefit sharing mechanism look like? At the moment, that is multilateral benefit sharing. And this is a bit of a sticky one for the Indigenous communities because it does somewhat take us out of the driver's seat into where our rangatiratanga or self-determination is. So it kind of is, how to explain, the multilateral system has a percentage or a bit of money from any commercialization of genetic resources or DSI. So you make a product, it uses DSI. You pay almost like a royalty into this big pool of money, which then goes out to different communities and different to support different benefit sharing options. So it kind of centralizes the benefit sharing system for DSI. However, the concerns currently from IPLC are who's governing that system, who makes the decisions of who gets benefits. And it's kind of like a nothing about us without us. So IPLC need a seat at that table to decide where those benefits go and how they're used. So those are some key points that are developing in the CBD at the moment. And it was a real pleasure to be there and to be advocating for Indigenous rights there. And it was really great to make a whole bunch of connections with the DSI community. So from there, we're going to talk about, Tim is going to discuss our survey that we did and the research we did into evaluation of DSI. Kia ora.
Yeah.
Thanks, Katie. We can stop sharing, that'll be great.
Stop sharing, I'll jump on.
Aroha, just a quick question. Dematerialized, what does that mean?
That's a good question. So it's another word for how it is separated from the genetic resource and DNA sample itself. So it's dematerialized as in it's digitalized. It's taken away from that. There's a separation between the digital version and the genetic resource. Would you say that's accurate, Tim?
Yep, that sounds good.
Kia ora.
Okay, so we're going to start sharing my screen. Let me get it up. Okay, so tēnā koutou, everyone. My name's Tim Coltman. I'm an emeritus professor at University of Waikato. Previous to my semi-retirement sort of role, I was the Dean of the Waikato Management School, and I've also spent a couple of decades looking at how to value products and services. And so the type of methodology we use is been implemented in this particular survey, and I'll talk a little bit about that as we go through it. So key issue is that there's political deadlock about whether the use of DSI should require benefits to be shared, and Katie sort of alluded to that already. Prior discussion and debate has focused primarily on concept and scope. So Katie used a couple of terms like fair and care principles, open as possible, closed as necessary. These are all the sort of topics of discussion, but there's been little discussion given to how benefit sharing might be enabled particularly in a multilateral type system, and what is the operational model that will enable that. We had talked about who will govern what and how the money will be shared, and are people prepared to forfeit money for a multilateral system? So these are pretty important questions. There's a strong need, we argue, for examples of how access and benefit sharing principles will be operationalized for any user of DSI. In many cases, scientists that are using sequence data like DSI are unaware of some of the policy implications that have been discussed. So to enable benefit sharing, we need an ability to predict what people are prepared to forfeit. So we need some economic model that sort of helps us understand how this would be operational. So how good are we at predicting the choices that people are prepared to make to enable benefit sharing? Well, we're not very good, basically. We've got 50 years of research on quality and accuracy of human decision-making reveals that we're quite poor. The process is inherently biased. And this is a bit of a tongue-in-cheek quote by Dave Parry here that illustrates the bias. I argue very well. Ask any of my remaining friends. I can win an argument on any topic against any opponent. People know this and steer clear of me at parties. Often as a sign of their great respect, they don't even invite me. So this person's clearly got an inherent bias about their own ability. The reality is there's no gurus, seers, specially endowed decision makers that can forecast how people are going to react and behave. Media love to report exceptions, that certain people are able to forecast quite accurately, but there's little evidence that they can do this other than random, and even less evidence that anyone can consistently maintain it. So we've got some challenges in trying to understand decision-making and what people will be prepared to forfeit and how much they're willing to pay for concepts like multilateral benefit sharing. So if we look at traditional marketing research, they rely on a concept called the concept test to test new products and services. What they do is get a small group of people together. They come up with a couple of guesses about the future and what the product might look and what features it might have. And then they ask them how likely you are to do X on a particular attribute. They come up with scores on 1 to 5 scales that are questionable, like what's a score of 4.3 really mean on a 5-point scale. The likelihood of forecasting success is likely no better than you or I. When was the last time you stopped to look at a particular asset or attribute in isolation to come up with a price or a decision to choose a particular product? Well, the argument is not really very often. What we look at is typically a combination of attributes and that reflect our decision to choose a particular product or service. So what does it take for scientists to share benefits and forfeit existing research budget? Well, it's not one attribute in isolation. You're not going to pay because the data comes from a particular database. You're going to pay because you've assessed a whole bunch of factors, like the level of access, how open it is, how controlled it is, where it comes from, what consent is being given, what level of provenance in terms of country of origin is known. And ultimately, from this data, you can calculate a willingness to pay concept. Once we measure what we call utility for access or consent or provenance, we can then calculate a dollar value on willingness to pay for a unit change in a certain attribute. So as consent level of consent changes, how much are you willing to pay to get greater consent, for example? The other challenge is that decision context matters. So there's many possible scenarios out there, and the scenario that you face, that you're situated in, influences what you're prepared to pay, obviously. So, if you have a $100 million budget, you might be prepared to pay more to a multilateral benefit sharing scheme than if you had a smaller budget. So what we had to do was come up with a scenario that we can test as a case example. And this is the scenario that we came up in our survey. We would like you to take on the role of a biology-based research scientist who has recently been awarded a $5 million grant to develop an aquaculture breeding program for green-lipped mussels. So you can see there's quite a lot of context in this particular example, and that's what we're we're testing. I'm not going to read through it all. You can probably see it later. We're running out of time. But this is a typical example that a research scientist who has a large grant would get a $5 million grant to look at this particular type of DSI use. And what we're interested in is how much of that money are you prepared to give to a multilateral benefit sharing scheme? So there's two types of data that we see in the world. So there's what we call revealed preference data. So this is data about real markets that exist in the world currently. The reality is we don't have a market for benefit sharing and multilateral agreements. So that data just doesn't exist. So it's not like you go and grant from the shop where you could test how much people are prepared to pay and what attributes they want. In this case, the market doesn't exist. So we need to come up with what we call stated preference choice models, where we observe responses made by consumers in hypothetical situations. And that's the situation I just alluded to before with the $5 million budget for green lip mussels. We developed statistical models that explain these situations. From these models, we're able to predict individual preferences and choices through a controlled experiment. What we're interested in looking at is the attributes and how people react to different attributes. So the manipulated variables in the experiment are what we call attributes. These attributes, to give them meaning, have levels. So we can define consent as a level of prior, so data integrity as a level of prior and informed consent. But that's pretty useless on its own. What we need to know is the levels of consent. So consent was awarded on specific use, it was awarded to general use, or there's no record of consent. So these are examples of levels. Same with data access. The definition of it is level of open access to data, but the levels can be fully open, controlled access, or could be an Indigenous repository. So what we're looking at here is how people react to these different attribute levels and measuring how much they're willing to pay for changes in those attribute levels. So this is an example of the experiment that that respondents were required to go through. So scientists and researchers make choices about sequence data by comparing and evaluating offerings at a more holistic level, where there's changes in consent, access, and provenance are all happening together, and it's by evaluating all that sort of combination in total that is what influences the choices that we make. So individuals do not choose a sequence data provider simply because it is one database. They look at multiple attributes. They make a choice when generic resources are involved by comparing different levels of access, different levels of consent and provenance. And this is the type of choice experiment they do. There's a statistical design that underpins this. They do 12 different choice blocks, we call them, and they evaluate which one they prefer. So that you look down vertically at option A and you look down vertically at option B and you make a choice about which one you prefer. And from that, we can measure utility for these different concepts and willingness to pay. So our sample was of 148 individuals, required them to make 1,600 choices. Approximately 82% of respondents were scientists. The remaining were considered policy advocates. The respondents had quite good experience, and the majority of respondents have experience with research project budgets. So that adds realism to the experiment, because we're asking people to evaluate a $5 million budget, and we've got the bulk of people have already considerable experience with evaluating research budgets. So they're the sort of people that we want to target, because they're the people that are going to be required to give up or forfeit some of their budget. When asked what percentage of total budget would you allocate to access and use DSI on genetic resources and traditional knowledge, the mean score was 123, but as you see from the graph, there's quite significant variance. One group, 15%, were prepared to pay more than $300,000. A smaller group, 10% or 11% there, were prepared to pay nothing. But then you see the variance here. So when we look at the results of the choice experiment, what's important in the type of method is that utility which is a preference you have, is a relative measure. It's only differences in utility that matter. So it's utility relative to something else that is important here. So if you think about the results here, we looked at consent, all right? So the reference level for consent was no record of consent. So if no record of consent exists, how much are you prepared to pay to actually get general use consent or specific use consent? And these are all the stars are all statistically significant results. You can see the size of the budget that people are prepared to pay to access specific consent. All right, so these are important results that help us understand how to operationalize model the benefit sharing concept, because we can see that people are prepared to pay. And this willingness to pay would underpin any future governance roles, etc. So we see the same for provenance. So there's no known, so the country of origin is not known. How much are you prepared to pay to know a known IPLC or a known country of origin? You can see the significant values here. What's interesting here in terms of access is that fully open access is not significant. All right, and so why is this the case? Well, we did some follow-up analysis and we identified there's two groups or two segments that explain the heterogeneity in this particular group. There's one group that comprises 73% of the population unwilling to sort of pay for open access relative to Indigenous repository. So the question here is they prefer an Indigenous repository, and if given fully open access, they actually have a dislike for that and are less willing to pay to a significant amount. But there's another group, 27%, are prepared to pay quite significantly more money for open access relative to an Indigenous repository. So it's dealing with this heterogeneity that is important as well in explaining differences in the group. Not everyone's the same in terms of open access is the bottom line. In terms of outputs, we've got a new model technique that's not previously been used to look at benefit sharing. It gives us a new understanding of the amount of money shareholders or stakeholders are willing to forfeit. We've written the start of an empirical paper for Frontiers in Genetics, and we'll continue to write conference papers and symposia presentations. So I think that's my time. Open to questions later, I think.
Yeah, kia ora, thanks, Tim. People feel free to chuck in questions in the chat as well if you have any that pop up. I'm going to share my screen here now. So just wanted to, yeah, so I had this kind of, we decided to use the context of digital sequence information as the one to explore this idea of valuation, partly because it was in the middle of thinking about how benefit sharing would come about, and therefore you had to say what value would it place on it. So I think this, this information from Tim is really interesting in terms of what people are prepared to do if the right information is in place. We know from what Katie was talking about that actually only 6% of samples have a country of origin associated with them. And therefore, there's a huge amount of material that is around that doesn't have that sort of provenance information that connects back. And so this is where I'm going to talk a little bit about what we've been doing with the biocultural labels through local context. And it's really been about how do you add information to the records that can reflect things like provenance, where the cultural authority sits, what sorts of protocols are in place, at least in terms of whether consent is verified or not verified, and then what kinds of permissions can be put in place as well. So really sort of stating the preferences of those communities that can sit alongside the record that's circulating in these open environments as a bridge into the next step in commercialization. So as I mentioned before, these a range of labels. We group them across provenance, protocols, and permissions. And this just gives you a sense of what they are. They have a visual component, the icon, which remains stable. And they'll have some text associated with it, which can be customized by particular communities. So I don't want to talk much about the labels themselves, but this example that's been put in place alongside Whakatohia. So great to see all the Whakatohia colleagues turning out today. And this is what happened in terms of some Whakatohia labels on Manaaki Whenua's collection database. Now, you can see there the five of the national collections which Manaaki Whenua is responsible for. So that's the essentially all the physical material, but the records about them have just been digitized into this new systematics collection database. Within this database, there's over 670,000 records. And at the moment, we've got notices, bicultural notices, which are disclosures on each of them, on each of those records, and over 2,000 labels, or labels on over 2,000 records. And this just gives you an idea of the numbers. So a notice has been put on each of the collections. And so while the notice is associated with each of the collections, it appears on all of the records in that collection, and you can see, the differences there. And then Whakatohia and Ngāti Maruki Taranaki have identified labels which they have put on the records that were collected from their areas. You see Whakatohia has done 1,200 records, and Ngāti Maru has 860. This is an example of what you see when you filter the results on the collections database, and just look for those that were collected from the Whakatohia rohe, and you'll see that represented there in the map view. And here's an example of one of the records which comes up from that 1,200. And you can see the Whakatohia labels that are sitting alongside it. There's a link to the biological specimens housed at Manaaki Whenua and Te Rohe o Te Whakatohia. And if you click on that, it clicks you through to the local context hub and provides information. That's the information that gets provided to the community for them to decide which labels they want to attach to the project. And this is what that looks like once that's been done. For each of the labels, if you click on the label on the database, on the systematics collections database, it highlights the customized text that the community have put in place as well. And so this becomes a way of making sure that the information which the communities provide is visible to any user, any viewer of the database. And the use of the labels is enabled through an API that works between the local context hub, where the communities customize their labels and decide which ones to apply to projects, and the research institution through the use of a unique project identifier. And one of the reasons we've tried to move into this more automated approach to this is that as data moves into those open spaces, it shifts very quickly from communities into institutions, across into aggregators, and then anyone else that starts to access and use that data. And then it may find itself through other activities and publications. And so across the data ecosystem, really trying to think about ways in which that provenance can be maintained, so that while there are some uses which communities will approve around, say, research or outreach where limited benefit sharing might be expected, there's other commercial outcomes where they might like to be involved in directly. And those are a couple of the labels that Whakatohia put in place around being open to collaboration and open to commercialization. So kia ora. I'm going to stop sharing there. And hand it over to Jacob.
Hello, hi. My name is Jacob Golan. I was previously a biologist, and now in the last few months of becoming a lawyer. And really, where I come to this project is, I guess, leveraging my technical background in operationalizing and making tools for these labels that Maui just presented on, taking two sort of approaches. One is sort of the ex ante preemptive role, where how do these labels get appended to DSI, and what is sort of the decision process that goes into which go on to a particular specimen, and then sort of the ex post enforcement or policing or even accessing of these data after the labels have already been added, or adding further labels to an ongoing project. And so really what my presentation will be on is focused on just very simple tools that will interface with the hub, the local context hub that Maui just mentioned, pulling from the API these labels and in various ways adding or calling them from different DSI specimens. So starting with the first, this is just a schematization more or less of what the decision process might look like on the back end, and to some extent been already put in place on the hub. It's small, but just to show that there's a variety of BC labels, and through a series of questioning, local communities or even just researchers can go through either as a familiar user interface or somehow on the back end, answering a series of questions to which yes or no would decide whether or not a particular label might be appropriate for a particular project or a particular specimen. And so taking, for example, just I like to use the case of rooibos tea, just as a classical genetic resource with associated traditional knowledge that has been misappropriated, reappropriated, and the whole story behind there. I'm sure many are familiar. And so what this might look like for let's say a particular community in South Africa, you'd go through this decision tree asking, is this resource limited to one community? Yes, and that would trigger some sort of decision in the chain to append these different labels, where it would be yes, and this label would be called and immediately associated with this particular resource. And when there's no, for instance, a particular label wouldn't be appended. And so you would just go through the whole tree, and then at the end of the day, you would have something that would look like, I'm skipping ahead, but that would look something like this on some sort of, front-facing public website, indicating all of the labels that would be associated with a particular collection, a particular genetic resource, or just a particular resource associated with a particular community. So that's fine in the case of something a little more tangible, such as, tea leaves, or in the case of rooibos, I believe it's bark. But what about when it's DSI, right? It's a intangible product, more or less. It involves an obscure string of nucleotides or protein sequences, and so that's where this idea of what does the underlying genetic code look like, whether it's DNA or protein. And so the typical format for DNA it's just called a FASTA file. It's the most basic file format. And just for an example, this is what it looks like. It's just a text string. And what's of key importance for use of the tools that I'll be demonstrating is this header line at the beginning, which up to the user can indicate all sorts of information. Usually, here it says SSU. It's a particular you could think of it as a barcode, more or less, of a particular species that allows people to distinguish, how I have this sequence of DNA, how do I know it's from what species? And so there are different regions in the genome that are useful for that, but also, more, I guess, mundane information such as latitude and longitude or acquisition ID, things of that nature. And so mainly we'll be working in the context of DSI with these types of files, this type of data information. And so then the question arises, if they're just these text strings, how then do we go from that to, bringing the user or the community wanting to use these labels? How do we connect the two? And this is especially important in the context of, how these digital, genetic resource, these DSI are presented to the public, usually in the United States. I'm speaking right now from New York. I'm familiar with NCBI, or the National Institute of Health, has this large database where researchers, second nature, often publishers, require it that you upload the sequences that you find so that they're publicly accessible, and the way they're found is through this header. And so that's what's, facing the public, whether or not they know that it's associated with a particular community, and then it might have, certain propriety interests involved is another matter. And that's where the second part of the presentation comes in. But just to say that these headers could have all sorts of strings of information, including some sort of contractual mechanism that would then lead a user through the header to some sort of click wrap or some kind of user agreement, similar to, if you're using a software, you click, I agree to the terms and services. And so all sorts of information are possible to present in this header, but the issue is, what's practical and what's feasible, and how can we use this mechanism already in place, the BC and TK labels that Maui just spoke about. And so that's where the R package that I've developed comes in. R is just a statistical language that's fairly user-friendly, and in creating this package, the goal was to make it as user-friendly as possible. There's no, very in-depth coding necessary to use it, and it's really meant to allow for easy interface between this local context hub, which houses all of the notices, and just the underlying data itself, how to take, a project, its associated BC labels, and associate them with digital sequence information. And so, not to worry too much about, the technical code on the screen. More so for our purposes, I'd say this schematic of how the package works is really what's of interest. And so it starts more or less with, you're a user and you happen to have a bunch of sequences. On the one hand, you could be, a local community who's recently taken an inventory of, let's say plants on, your native lands, and you want to, and you have now all the DNA barcodes associated with them, and you want to, preemptive kind of defensively secure the IP rights or prevent others from usurping the IP rights by making already public that this is what's at hand. Or on the other hand, you could be a researcher who wants, to engage with everything that we've been speaking about previously, and you want to associate these resources with the particular communities on whose land you've been conducting your research. So you start with the sequences. Then from there, you can, through this R package, which, by the way, it's called Enrich, after a lot of the initiatives I've been involved with, but capital R for the R programming language, and so through this package, you can then search the hub. So you can search the local context hub, see all the projects that are there, and see which might, let's say, you could ask, what are all of the projects that have been uploaded to the hub for South Africa, for example. And you can search and it'll populate everything. It'll make a call through the API and it'll populate, all of the projects that would be of interest to you. Starting again, or using the example of, you're a researcher and you have a bunch of sequences and you want to associate a particular string of labels, or there's a particular string of labels with a DSI, you could then fetch and append a certain metadata associated with that project using this unique project ID, which Maui mentioned. So each project in the hub has just a random sequence of numbers that are associated with it, and automatically link what labels are on the hub and associated with the project with a particular digital sequence. And then from there, you can attach it to this sequence header, this DNA sequence header. You put the unique ID, and from there, all the labels that are associated with the project on the hub automatically are now associated with a particular string of DNA. And you can go back and forth, right? You can take a sequence that has the header already in place with the unique ID and use it to search in the database, in the hub, to see which labels are already associated with it. And so just to go into a bit finer detail, so it's very simple. Like I said, I tried to make this, this tool as, no in-depth knowledge of R necessary. You install the package with a one line of code, and you type the command index all projects. And at the time that this slide was created, these were in the initial days, so there were few. And this populates all of the current projects that are in the hub. And you can see you get the results. You have these unique IDs, which are just the random string associating projects to their labels and notices, the title of the project, whether it's public or discoverable, certain privacy settings that are in place, and also the date added and the date modified. And so you can just right away access the entirety of the hub through this very simple tool. But then from there, let's say you don't necessarily know what you're looking for, or by now I'm sure the hub has many hundreds of projects, let's say. Maybe you have a piece of information that you want to use to hone in. So you can use the command find projects, and you can search by any one of these headers, basically, that are already in the API structure. So you can search, the title of a project, but you don't necessarily know which labels are associated with it, or a date range, etc. And any combination of the above. But really, what is what links specific projects to the relevant labels is this command find projects ID, and that allows you to look up very directly. Let's say you go to some kind of DSI repository like NCBI that I mentioned, or whatever other ones exist. You see, oh, there's this unique header. There's this unique ID in the header. I need to look up, what are as a researcher using these sequences, what restrictions are in place that would be provided to me through the notice of the BC labels. And I can look those up by simply searching in the header, which is a very, simple, it's easy to extract the header from a sequence, and you can easily interface with the hub and search the unique ID. And once you're there, it will tell you all of the names. So once you're there and you've searched the unique project, you can see all of the labels. You can download, you can see what there's text description, which can be customized per project or per community, but also the digital, the image itself of the label can be accessed and downloaded through, a few more steps in R. But also what this allows is a way of not only, accessing it as a researcher or as a researcher uploading the sequences, or as a researcher looking to see what notices are associated with a particular set of sequences, but also this allows for an easy, calling from one platform to the next of the BC labels associated with a project. So let's say, it's you have the hub, and you have, your lab's website, and you say, these are the sequences associated with our past research. This would allow you to very easily pull between the two, so that you have this, on the one hand, local context hub, which is just a repository for all of the DSI and BC labels and, all the projects associated. And then on the other hand, you can just pull in very directly. So it prevents kind of duplication of efforts, and it allows just for this information to very simply be called by really anyone who, in 15 minutes wanted to become slightly familiar with R. And I think, yes, and it's called Enrich, just like it sounds, with a capital R. I can share in the chat the GitHub. Thank you.
Thanks, Jacob. So, hey, look, so yeah, you can see there's these sort of two different components that we've been working with as a part of this project. One that really sort of thinking about how you think about the value that the data will have, and how that would then contribute into benefit sharing arrangements. And the other component, which has been more about how you might track where the data is going, so that you could know when it needed to be subject to benefit sharing as well. So look, we're open for questions. Feel free to click your videos and join us in the room and ask away.
Hi, everybody. I'll jump in because I have a question for Jacob. I love thinking through how to integrate the labels into the actual data files themselves. And I was just wondering, so I'm going to play devil's advocate, and it's probably true, of being a completely terrible researcher utilizing an open digital repository. Okay, so very naive, never thought about the labels. And all I'm doing is downloading a specific taxonomical group of species, and I'm kind of agnostic as to where that comes from. And so the first usual step is kind of a cleaning, and then you might align them to each other. And there's multiple tools that you would then work on the data with. And multiple of those tools actually have a maximum FASTA header character length, that if I naively, my first port of call when I get like, oh, error, your FASTA header is too long. I do a head, file name, and then I say, okay, how can I cut that? And I don't even necessarily, traverse the whole file. And then suddenly, if your file isn't in the top 10, if your header isn't in the top 10, I'm completely removing straight away your accession from all naively, not on purpose or out of malice, but that will impact when I do my alignment, that information is then lost. Is that something that you've thought about when you're developing your tool? And just super interesting and cool tool.
Thanks. Yeah, it is. It was something we discussed a lot. Like, it's just a text file, a FASTA file. What's, and even a bad actor, playing the, real devil's advocate can just erase the unique ID. So it is a problem, and there's no, it's true of any FASTA file, any of the information. And so luckily the unique ID, I believe it's only, 16 characters. It's not incredibly long. But at the same time, implementing them in the first instance is at the the goodwill of the researcher. So if it's really a priority, even if an alignment program doesn't allow you to have a header of X length, it'd it's, it presumably would be on the good actors agenda to reappend the headers, which I've had to do for other reasons anyway. So on the downside is, yes, that it's somewhat inevitable that there's no way to avoid without changing the underlying data structure. But these headers are easy enough to modify that they can just be easily reappended for good actors.
Cool, thanks.
Peter.
Ai, kia ora no tātou anō, te whānau te whakatohea, tihei moriora. Ko Petra Hudson tēnei no te whakatohea tūturu, working out of Massey University on the identification of Māori epistemologies and ontologies that can be written into algorithms. Question goes to Tim. Kia ora, Tim. Within the economic model or modelling that's been taking place within the work, whose model is being used? I.e., is there an Indigenous model that is being used within the economy existence? Because certainly when I look at a European model of economy, it's extraction, eradication. Whereas in a Indigenous one, we tend to have a kaitiaki type of modelling. Has that been trialled at all? Ngami.
So, yeah, it's a tricky question to answer that one. I'm not quite sure how to answer it, but let me have a go. At the end of the day, the model only speaks to the domain that we are interested in. So the domain that we look at is that scenario that we presented, where it was around green lip mussels and you had a 5 million, and the attributes that we look at around provenance, consent, access, that's the domain. So we can only speak to issues around those issues. Anything else is outside the model, outside the domain of the model, and we can't speak to it. But that's to some extent, the power of the model. It's very, I guess, comprehensive around the attributes you look at and the scenario you look at, and that's the case. I guess it's a case example of that particular scenario only.
Nice. I understand. I understand. So thank you for that.
Can I just add? So I think so one of the things when you look at the Convention on Biological Diversity is that it actually talks a lot about conservation. And so, kind of the purpose of the convention is to support biodiversity and it sort of speaks a little bit more to the kaitiakitanga focus, even though it's kind of more of a Pākehā one and not necessarily an Indigenous one. The thing is, it's in some ways it sort of fudges the fact that there is this commercial enterprise that it's also supporting. That kind of protocol is also supporting, and there's a lot of value that's generated off genetic resources being taken and used from Indigenous communities without really any mechanisms for how that gets shared, how that value gets shared. And so that's why we've sort of probably dragged it away from the big kind of different way of the whole economic model to within the economic model as that income's being generated, what are the ways in which value should be passed back? And this was really just trying to get a sense of what would people be willing to share? What would they think is fair as a researcher for access to that data?
Yeah. Kia ora. Thank you, Tim. Thank you, Maui. You understand.
We tried in terms of selecting the attributes to think about that, like what were the things that were important. And Jason Mick has been a part of those conversations as well. Yes. So to try and just see where those overlaps, some of those overlaps in thinking might occur.
Cool. My issue is the compromising in between the two and the where you're going to. The marginalization is going to still happen with the Indigenous people. but no, understand where you're taking your modelling and your trialling through. Because obviously, as another model, we could just go Tūturu Māori and work it that way. But I just wanted that. The other thing I just wanted to ask Jacob, whose databases are you using to run your sequencing through?
So right now it just relies on whatever's in the local context hub. It doesn't depend on any one's particular database. It's for instance, if a user has their own just in-house database, these are the specimens that I've collected.
Right.
I want to add the unique ID to them. It allows for that, but also it could, using other packages and tools, it could pull from any database that you want.
Nice. So it doesn't have to be big data. It can be
one
data piece.
Yep.
Yeah.
Mm-hmm.
All right.
It's as you might see, Corey put a note in the chat saying there's at the moment 122 projects on the hub. So we're not dealing with big data yet, but conscious that as it gets used and applied more generally, then those things need to happen. And so being able to create ways in which parts of the process can be automated as much as possible become important. And even if you start thinking about benefit sharing for sort of access to data and whether royalties and things might be applied, and whether you can start doing sort of attaching smart contracts to particular labels that then support some kind of royalty for access to the data. There's sort of a whole range of more kind of functional tools that might emerge in the future. And I think once you get into that part, then you're really heading down what Jacob was saying about in what ways can the access and use be more enforced. So any other questions? I say we've kind of hit the top of the hour.
Apology. I didn't realize we had to turn our cameras off, and I thought
all.
I was just bowling into the marae and I could see everybody. And when I was picking my nose, I just apologized. But sorry for keeping my camera on, Hānuere.
Again, just showing the world we used to have. We're used to having sun here as well, not just rain. All right. Hey, well, kia ora koutou. Mihi ana ki ngā kai kōrero, Katie, Tim, Jacob, thanks very much for being available this afternoon, sharing some of your work with us. Just to everyone here, we know us as well as Petsera. This has been recorded and will be up on the Tikanga Technology website under the Whāki webinars. So everyone else that wasn't able to make it because of the time of the day or competing things will be able to look at it as well. So just thanks to you also for being available, and hei whakakapi ngā kōrero tēnā hi kia tau kia tātou katoa, te atapau te tātou ariki kaiti, te aroha o te atua me te whiwhinga teitanga ki te wairua tapu ake ake. Āmene.
Āmene.
Kia ora koutou. Thanks, Neil.